All right, I think we can go ahead and get started. So hello, everyone. Thank you so much for joining our IBEX workshop today. I'm really excited to have Aanandita Kothurkar from University of Sheffield and Ola Krzywanska from UCL here with us. So Aanandita and Ola will be talking about their work and offering some tips for using IBEX and zebrafish and other animal models. But before I pass it off to both of them, I do just want to share some slides introducing Protein Tech in case you have not heard of us before. So, Protein Tech is a leading manufacturer of antibodies and immunoassays. We have a really expansive portfolio of antibodies that encompass over 13,000 targets and many different research areas. We also offer immunoassays such as ELISA kits and IHC kits, and we also offer other kits like cell separation kits and antibody labeling kits. Within our Protein Tech brand, we also have our humankind branded cytokines and growth factors, and all our proteins are expressed in human HEC293 cells, so you can be confident that they have the native folding, post-translational modifications, and glycosylation that you would expect to see in human proteins, and that ultimately increases their bioactivity. The third line that we have is our Chromatec branded nanobody-based reagents. So nanobodies are about 10 times smaller than traditional antibodies, and they can bind to their targets with really high affinity, making them ideal for clean immunoprecipitation, super resolution microscopy, as well as protein purification. And then lastly, we also have a subsidiary of Protein Tech known as Protein Tech Genomics. And Protein Tech Genomics manufactures antibodies that are conjugated to oligonucleotides for single cell RNA sequencing. As a company that is driven by scientists, we are here to support our researchers and scientific community any way that we can. So, our award winning products are self manufactured, and we offer the largest in house portfolio of antibodies for the human proteome. We also know the importance of product validation and offer open access validation data. When testing new products, we validate with relevant sample types, appropriate experimental conditions, and using knockout knockdown models whenever possible. Also, as antibody experts, we have in depth knowledge of immunoassay applications, and we can offer outstanding technical support for our customers. We also stay on top with cutting edge technology, such as our proprietary antigen specific B cell cloning and engineering platform that we use to manufacture our UniRab recombinant antibodies. With 7 inventory sites and manufacturing and research sites on 3 continents, we are able to offer next day delivery to many areas in the world so that you can get started on your experiments right away. And as a company, we do measure our success by our customer scientific success, specifically by helping them publish impactful research faster. So, this graph on the left is showing that over our company history, our products are featuring in more and more publications each year. And as of now, we have over 300,000 publication citations, making us one of the top 5 cited antibody companies and also one of the fastest growing companies in product citations. I also want to highlight that as a company, we strive to make the searchability of our products on our website as user-friendly and informative as possible. We've recently launched some new updates on our website to help our customers select the right antibody. This includes our unique 3D epitope viewer that allows you to interact with epitope structures and explore antibody binding sites so that you'll know exactly where our antibodies bind to your protein of interest. We've also introduced advanced filter options to help you find exactly what you need, such as our immunogen region selector toggle that allows you to filter for antibodies generated from specifically your region of interest. Additionally, you no longer have to filter through all the variations of an antibody from the search results. You can view all formats of an antibody, including conjugations or any sort of buffer formulations, on just one page. And this ultimately will streamline your process for antibody selection. So with that, I would like to introduce our speakers for today. I'll start off with Aanandita. So Aanandita is currently a neuroscience PhD student at the University of Sheffield studying compartmentalized synaptic plasticity in Drosophila. During her work as a research technician with Ryan McDonald at the UCL Institute of Ophthalmology, she adapted the IBEX multiplex immunofluorescence technique for use in zebrafish and novelly combined it with in situ hybridization and whole mount immunofluorescence. I also want to introduce Ola. So Ola will also be joining us today. So Ola is a PhD student at the UCL Institute of Ophthalmology in Dr. Ryan McDonald's lab. Her project aims to establish the killifish, a rapidly aging teleost with a lifespan of just 6 months, as a model for studying retinal aging. In her project, she is utilizing the multiplexed immunohistochemistry technique IBEX to investigate cell-specific changes in the aging retina. So welcome, Aanandita and Ola. Thank you so much for being here. And I'm going to go ahead and pass it off to you guys. If at any point you guys do have any questions for Ola or Aanandita, please feel free to input those into the chat or the Q&A box, and we will address those at the end. So I'll go ahead and pass it off to you guys.
Thank you so much for having us. Let me just share my screen. So, hi everyone, I'm Aanandita, and today Ola and I will both be introducing you to the IBEX technique to revolutionize your multiplex immunofluorescence. So this is the agenda for today's talk. I'll be introducing you to what IBEX is and understanding the workflow of the experiment, and then how we adapted IBEX for zebrafish, as well as combined it with a modified form of in situ hybridization, which is HCR. I'll then pass you over to Ola to talk about adapting IBEX for unconventional animal models, as well as how to best implement IBEX in your own lab and some common troubleshooting issues that we faced, followed by any questions you guys might have. So, immunofluorescence is a widely used technique that's used to study the gene of your interest by looking at where the protein is expressed. And the way that this is done is using a primary antibody that binds to your target antigen in your tissue, and then a secondary antibody which then binds to the primary antibody and fluoresces, allowing you to visualize your protein expression. And in this experiment, you have your secondary antibodies, which are host specific to the host that the primary antibody was raised in. So if you have an anti-rabbit secondary antibody, it will then bind to every rabbit antigen present in your tissue. As you can imagine, this creates problems as you can't use multiple antibodies raised in the same animal, as any secondary you use will bind to both of them and you won't be able to differentiate between the signal. Therefore, traditional immunofluorescence experiments are limited to 2 to 3 antibodies per tissue because multiple commercially available antibodies are all raised in similar hosts. And traditionally available microscopes like the confocal, which most labs tend to use, are also limited in number of channels that you can image at a time. So this restricts your experiments and creates challenges, especially in use for studying immunology or wound repair and regeneration, where you would like to look at multiple proteins at the same time, as well as anywhere where you want to study simultaneous interactions between more than 3 different types of cells. Particularly in our lab, we wanted to look at multiplex immunofluorescence to do things like localized cell death when we do like tunnel assays, to look at which cells specifically may be dying first. Also to characterize mutants after CRISPR microinjection or adult tissue, anywhere where we have limited tissue and we want to maximize information that we can gain from it, and to overall reduce the number of animals we use in our experiments. So a solution we thought could work for this was IBEX. So IBEX stands for Iterative Bleaching Extends Multiplexity, and it was developed by Radke et al. A few years ago in mice and human tissues. And it's a multiplexing immunolabeling method that allows you to visualize several different antibodies on the same piece of tissue. So as you can see in that image here, they visualized the 60 different antibodies. And the way they do this is by conducting sequential rounds of immunofluorescence. Each round contains 3 or 4 different antibodies and then is separated by a round of bleaching. This bleaching quenches all of this fluorescence from the previous round and gives you a clean slate so you can visualize the antibodies in the next round. And this is the IBEX workflow that we adapted to work for zebrafish. So the first part is having an adhesive coating on your slide. So obviously with the IBEX protocol, your tissue is going to undergo multiple rounds of staining and a cover slip application, cover slip removal, and this can be quite harsh on the tissue and the tissue might fall off your slide. So therefore we use adhesive coating of chrome alum gelatin on the slide first, and this helps the tissue to adhere better to the slide. The next part is a crucial part of IBEX, which is the conjugated antibodies, and is also an important change we made to the original protocol. So the original protocol usually used direct conjugation kits, but we used microconjugation kits. And a conjugated antibody is simply when your primary antibody is directly linked to the fluorophore. So it takes away the problem of the secondary antibody recognizing multiple different antigens, as there is no free fluorophores floating around to bind to other antigens. And it allows you to use multiple antibodies raised in the same host together without any interference. So once you have these conjugated antibodies, you start with staining your sample and imaging it. After that, you bleach it using a solution of lithium borohydrate under harsh white light, which also helps to reduce this fluorescence. After that, you can restain with your next panel of antibodies and repeat this cycle until you've stained for all the proteins you would like to visualize. Once you've gathered all these different images, you can do registration based on an alignment software. We'll discuss this in more detail later, but it's based by aligning a single channel that's present through every single round of imaging. In our case, it was DAPI, which is a nuclear stain. And based on these, it can combine all of your images into a single image, and you can visualize all your antibodies together. A quick note on the conjugation of antibodies. So we use the flexible protein conjugation kits by ProteinPack, and this is mainly because they allowed us to do microconjugation. So there's a few other conjugation kits available on the market, but they largely require you to conjugate quite a large volume of antibody, and this may not always be feasible for labs to convert their entire stock into a directly conjugated antibody or to purchase these antibodies. With the flexible kits, you can have the flexibility of also conjugating your antibody to different fluorophores. So for one experiment, if it works better for your antibody to be conjugated to an Alexa 444, but another one you want it to be conjugated to Alexa 647, that's possible with flexible. However, if you prefer using directly conjugated kits or commercially available conjugated antibodies, those are also compatible with IBEX. So this is just the workflow of flexible conjugation, where you mix 0.5 micrograms of antibody with flex linker and flex buffer. You incubate them and then add a quencher, which binds to any leftover fluorophore and prevents that from them binding to the anything in the sample. You incubate this, and then your antibodies are ready for use. A quick note on this as well is that when we adapted it for zebrafish, we found that doubling the volumes of each component of the reaction worked best for us, but you can try it out yourself and see which one works best for you. So we adapted IBEX to the zebrafish retina, which is the tissue of interest in our lab. And just to give you an idea of the images you're going to be looking at throughout this talk, this is the structure of the zebrafish retina. It has 5 main neuron types, so the photoreceptors, the horizontal cells, bipolar cells, amacrine cells, and retinal ganglion cells, as well as one main glial type, which is the Mueller glia. And we have robust antibodies to label each of these cell types, which we combined when we adapted this technique for IBEX. So this is our image of 11 different antibodies used together on a single piece of zebrafish retina tissue. And this was done over 4 different rounds. Most of the round contained 3 rounds of antibody and 1 round contained 2. And over these 4 rounds, you were able to successfully label every major retinal cell type and visualize them together in a single picture. After this, we wanted to check whether we could combine IBEX with our transgenic fish. So transgenesis in the fish is fairly routinely done, and there's a wealth of lines if you want to label your gene of interest with a fluorescent protein. And this is particularly important because sometimes these transgenic lines can give you morphological information that can be lost with antibody labeling. And here's an example of just some of these lines that are available for the retina in particular. So we wanted to see whether these lines could be combined with IBEX as well as bleached so that we could conduct rounds of IBEX on top of these tissues. So this is what we investigated, and we looked at most of the GFP lines that we tried did bleach, but not with lithium borohydrate. So we had to, when we do traditional immunofluorescence, we tend to use antigen retrieval, which is when we boil the slides in sodium citrate or tris HCl in a Copland jar for about 20 minutes. And this naturally quenches the transgenes, and then we usually boost them later. So we took advantage of this, doing this for IBEX. So in the first round, before we started the immunofluorescence, we would quench the transgene with antigen retrieval, then conduct the IBEX with multiple rounds, and in the last round of IBEX, boost this transgene again with an antibody against GFP. So this worked pretty well for our lines that had GFP, but we also tested lines that tried RFPs or YFPs, and these did not bleach even after harsh antigen retrieval treatment. So we concluded that you can combine IBEX with transgenic lines, but you'll have to test these on a case-by-case basis. Even if your line of interest does not bleach, that doesn't mean you can't combine it with IBEX. It just means that it will occupy a channel in each of your rounds, similar to the way DAPI would. and you can actually perform alignment with that one as well. So after that, we wanted to combine IBEX with a modified type of in situ hybridization. So particularly in zebrafish, if you have a gene of interest, it can be difficult to find a commercially available antibody that works against this gene. So a lot of people resort, especially in the last few years, to in situ hybridization to look at where your mRNA of the gene is expressed. And the HCR, or modified in situ hybridization chain reaction, basically functions by having a probe sequence that binds to your target mRNA, and you can think of that as a primary antibody binding to your protein in your tissue. And then, similar to secondary antibody, you have hairpins. And these hairpins bind to the probe, but also are complementary to themselves. So they bind to the probe and then to each other to create this chain reaction, which actually amplifies the signal and gives you much better fluorescence. So when you do this experiment, sometimes it can look like this, and it's clearly localized to a single cell type, which in this case is the mucoglia. But it can also look like this, where your RNA is spread across multiple different cell types, and you're not sure exactly where your RNA is being expressed. So we thought we could solve this problem by combining HCR with immunofluorescence by bleaching the HCR signal initially, and then conducting a round of immunofluorescence on top of it, and if we used antibodies that we knew were cell-specific, and if we could observe overlap, we'd be able to localize this RNA expression to different cell types. So this is what we did. We did a first round of HCR against 3 different genes. So CYP26A1 and GluLA in cyan and red, which we knew would localize to the myeloglia, and VSX1 in purple, which we knew localized to bipolar cells. We then did a round of bleaching, but I should mention for the Alexa 555, which is in red, the bleaching was not sufficient to get rid of the signal, and we had to do a gentle type of antigen retrieval, which was heating the slide in sodium citrate at 60 degrees Celsius, and this then got rid of all the signal. We then did a round of immunofluorescence where we looked at 2 different antibodies against that we knew localized to myeloglia. So in yellow and pink, the carbonic anhydrase and glutamine synthetase, and then 1 antibody that was expressed in bipolar cells, which is the PKC beta. We then overlaid these 2 slides and observed the overlap between the stainings. I've shown the one for myeloglia here, but if you would like to look at the one for bipolar cells, it's available in our paper. So here you can very nicely see that the antibodies in yellow and pink are overlapping with the RNA expressed in cyan and red. So it's a very useful technique if you would like to identify and localize this expression pattern of your mRNA. So that's all for me, and I'll be passing you over to Ola to talk to you about adapting IBEX for unconventional animal models and some troubleshooting.
Thank you so much, Aanandita. Hello, everyone. So the work that Aanandita presented so far has been done on the zebrafish retina. And in our lab, we also wanted to expand IBEX to some other models, more unconventional, to show that the method can be applied to many research organisms. And we have tried IBEX on retinal samples from African clove dog, a frog, and African turquoise killifish. Killifish, as some of you might already heard, is a rapidly aging teleos with a lifespan of merely 6 months, what makes it an exciting model for aging research, especially for my PhD project, as has been mentioned at the beginning. And such rare models, however exciting, are still holding some limitations. Because they are relatively new, their husbandry is still a challenge, and because of that, the availability of tissue for experiments. Something happened.
Yeah, it seems like Aanandita got kicked off, so hopefully she can hop back soon.
And in the meantime, I have the presentation as well.
Perfect.
Sorry, didn't expect that. Just give me 5 minutes.
Something always happens. It's all good.
Um,
She is back.
Okay, I can share it anyway since we've been not connected. I'm just re-downloading it because we had it on SharePoint, and I don't have the.
Sorry, I'm back.
Aanandita, okay, that's fine. If in case it happens again, I now have it, so we can I can reshare it. Sorry, everyone, we've been interrupted. I was just saying that the models are very exciting indeed, but they still are holding some limitations because they are relatively new. Their husbandry is still a challenge. And because of that, the availability of the tissue that we have available for experiments is extremely limited, and each section has to be used really carefully. Moreover, transgenesis, which Aanandita mentioned to be a technique widely used in zebrafish models, is still quite challenging in killifish or the frog. And the number of transgenic reported lines is limited. This makes our area of research rely highly on immunohistochemistry methods and alternatively on in situ hybridization chain reaction, while whenever we want to label cell populations. So this is where IBEX comes handy and becomes very helpful by allowing us to maximize the information that can be obtained from a single piece of tissue. And it allows us to identify cell subpopulations that would not be possible with conventional immunohistochemistry methods. And just to show you an example, we have successfully adapted IBEX to killifish retina. And on the slide, you can see a ciliary marginal zone of juvenile killifish retina and different cell populations labeled on one piece of tissue with multiple rounds of IBEX. And a postdoctoral researcher in our lab, Dr. Nicole Noel, has also performed IBEX on the retina of African cloved frog. And in the picture, you can see IBEX done on a 5-month-old frog sample with also multiple different cell types labeled together on a piece of tissue. And I hope you can also appreciate how different each retina is. So now the part that can be the most useful for all of you after some introduction. Maybe some of you are already preparing to run an IBEX, maybe starting doing some plans. So hopefully after this webinar session, you will know more or less what it entails and what are the crucial steps of IBEXing. So if we could go to the next part. So what do you have to do to implement IBEX in your lab? The first step, although might see as a very simple thing, is antibody validation. You have to know what's your expected result for each antibody. You can't just throw the antibody onto the IBEX panel without first validating it, because then you don't know whether there is the weird result you might be getting, whether it's because your antibody just doesn't work, or is it because of multiple rounds of antibodies that are destroying the signal. Moreover, you should test if your antibodies work with conjugation kits, either standard conjugation kits or the microconjugation kits that we mentioned. And moreover, you have to test whether the signal of your antibody is quenched by bleaching, or maybe you need some harsher methods, such as a gentle antigen retrieval, which Aanandita has already mentioned. This will become essential for the last part, while you want to put everything together and you don't want other antibodies going giving you non-specific staining in different rounds. Once you validated all your antibodies and you know you want to start, you might proceed to preparing your slides. As Aanandita already mentioned, for IBEX, we usually section our tissue of interest on slides that were pre-coated with chrome alum gelatin. This gives us a better tissue adherence and prevents washing off the tissue while we bleach it and stain it, block it multiple times in multiple rounds of IBEX. You should also consider a right choice of a mounting medium. You should always choose for IBEX a medium that is water soluble and that is forming only a semi-permanent seal, so that you will be able to remove the slide after finishing your imaging, because somehow you have to reapply another part of antibodies. The mounting medium that we are using in the lab that worked really great for us is a Fluoramount G mounting medium, which you can see here on the picture, but perhaps you have something better that you think might work for you as well. This is just our recommendation. And what is also important in the entire process is a choice of a right cover slip. You should make sure that the cover slip that you're choosing is smaller than your slide. And as you see on the picture, the cover slip that is covering the slide is much smaller, is more or less a half of the size of the cover slip. This will be important in the step after imaging. When you want to remove the slide, this cover slip will simply have to fall off in a 50 ml tube, and if the cover slip is too big, it will simply take the tissue with it. So we want it to be as small as possible. Once you've done all these steps that I just explained, you might proceed to imaging. So after you apply the mounting medium, close it with a cover slip, within the 40 minutes, you should start imaging. Why 40 minutes? It's enough for the mounting medium to dry, so that if you put the slide on the objective, it doesn't damage the tissue by still moving. And we also want to do it as soon as possible so that the medium does not dry. So we found that 40 minutes, it's more or less an optimum. But if you have a bigger slide and you have to apply more mounting medium, then of course the time might differ. And as I already mentioned before, right after you're done with imaging, not 3 hours after imaging, not 1 day after imaging, but right after you're done with imaging, you should place your slide in a 50 ml tube with water or PBS to allow the cover slip to fall off. And this is the step where the size of cover slip matters. And please allow it to fall off by itself. Don't force it. Any force, any extra movement can cause the cover slip to take the tissue with it while coming off. And our recommendations is to image multiple sections on one slide, because some might wash off in the process of subsequent stainings. I know that when you're usually doing regular immunostainings and want to compare some time points, usually one image of each time point is enough. But for IBEX, we recommend to have a backup, because it's very unfortunate if your tissue comes off in a round 4 and you had 6 rounds planned. So always have a backup. It was very useful for us in the process. After you're done with the imaging, your cover slip fell off, wash it off a little bit and give it like 3 extra washes with PBS, and proceed to the bleaching step. So now you want to remove the fluorophores that you've been just imaging, and we do it with a lithium borohydrate. It's quite a toxic and explosive chemical. However, we usually just dissolve it in water under the fume hood very carefully. The small amounts that we are using and the small volumes have never caused us any issues, but being extra careful is recommended while dealing with the reagent. So you dissolve it, you wait around 20 minutes for it to activate. You will start seeing bubbles. And you apply it on a slide and place the slide with lithium borohydrate under direct white light. And ideally, you should confirm bleaching of your fluorophores under the microscope before the next round of staining. And in case you still see a signal, we recommend antigen retrieval methods, either with sodium citrate or TRIS HCl. And once all of your IBEX rounds are done, so the steps starting from mounting up until bleaching, all these steps will be repeated with each IBEX cycle. And after you have, let's say, 5 rounds of stainings, which can give you around 15 antibodies, you of course want to put them all together. And we do it with a simple ITK registration software, which is available to download on GitHub and has been described in detail in the Radke et al. Paper. You simply register all your confocal stacks based on one signal. We do it by the alignment of a DAPI signal because this is the antibody that we have in every round. But if you have some other robust antibodies, stainings that are present in each of your in each of your IBEX round, you can use this as a registration signal. And here is just the image showing that we have here, for example, 3 rounds. This is how the DAPI signal looks like after each of these rounds. And it looks quite comparable. And what the software will do for us, it will put it together with all the other antibodies in it, but only based on DAPI signal. So this was more or less step by step of how the actual process looks like. It's usually quite a lot of work, but what is also very important is that before you proceed to all this work, you should carefully plan and carefully design panels of markers that you want to use in IBEX based on your previous individual reactions and bleaching tests. So based on all the antibody validations and all the tests of bleaching, which Aanandita has showed you how the transgenic lines can be tricky. We also experienced some antibodies are more difficult to bleach. So this is all about planning. So why is that? Why is planning so important? It's simply because certain fluorophores are more amenable to bleaching than others. There are some more robust proteins on your tissue that will be more difficult to bleach, more robust antibodies that will be difficult to get rid of. There are some antibodies that you will realize in your testing phase that don't work well with conjugation. And these antibodies we usually recommend to use in the first round of IBEX, which is usually done with standard primary followed by secondary antibody reaction, but only if you want these primary antibodies to be involved in your IBEX panel. On the contrary, weakest markers should be used in early panels. So after you do your primary, secondary first round, and then you know what are your weak markers. You can use them in a round 2, because then it increases the likelihood of strong signal detection. And even though the images look beautiful after all the registration, each round of bleaching, each round of staining makes the tissue degrade a little bit. And in the later rounds, it's very difficult to make a weak antibody work well. And brightest and least efficiently bleach markers, such as lectins or transgenes, should be used in last rounds to minimize the potential for any leftover signal. So it makes sense. What is weak? What gives you a weak signal in your testing should go in the first rounds. Something that is extremely robust, you should leave for last rounds. This is also because, as I mentioned in the first part, certain fluorophores are just more amenable to bleaching than others. And what we have found, and what have also Radke et al. Found in their original paper, is that whenever we use some antibodies in the first round in the Alexa 555 or 594 wavelength, this can be very difficult to bleach, and we strongly avoid using strong antibodies in that channel because we have done problems with bleaching these antibodies with lithium borohydrate or antigen retrieval. And of course, you should consider your availability of species and fluorophore conjugation kits. Let's imagine you run an antibody, a rabbit antibody in 488 channel, and in the next round you have to use some antibody in rabbit, that is rabbit, but in 555 channel. So for each of these rounds, you will need another conjugation kit. So rabbit 488, rabbit 555. Usually all the companies, we use protein tech, so they are all available. You just need to purchase them separately if you want to use different hosts with different fluorophores. So in conclusion, we adapt the IBEX method for use in zebrafish, killifish, and xenopus. What allowed us to label multiple retinal cell types at the same time and visualize special relationships of cells in the retina. We combine this technique with the in situ hybridization chain reaction. And something that we didn't mention in this webinar, but what you can find in our paper, is that we also adapted IBEX to the Hall mount techniques, which is more of a 3D imaging. For us, it's an eye, but it can be also done on other parts of the body, which allowed us to expand the zebrafish toolkit. And we are sure that the IBEX will revolutionize our ability to visualize multiple cell types in any tissue, in any organisms where antibodies are readily available. Thank you so much. That was all what we have prepared for today. We thank our labs from Dr. Ryan McDonald, where all the work has been done, and the Lin Lab, which is the current lab of Aanandita. After Afrida gives you a short closing presentation, we are more than happy to take any questions. And if you have even more questions and some things are still not clear, you can always read to our paper that has been published at the end of last year, in which we describe adaptation of IBEX to zebrafish, killifish, and the frog. And we are also very happy to answer any questions by email. Thank you so much.
Great. Thank you so much, Aanandita and Ola. That was wonderful. You guys did such a great job explaining IBEX in such a simple way. Like, it sounds like such an easy process, but I'm sure there is so much troubleshooting and complexity that goes into it. So thank you so much for laying that out so nicely for us. As Ola mentioned, I do just want to share a few slides before we begin the Q&A. I see all the questions coming in. This is great. Keep them coming. We'll get to as many as we can. But before that, I just want to share a few slides to highlight some products that might be interesting for our audience today. So Aanandita and Ola, of course, have been talking a bit about the advantages of using flexible for IBEX and just multiplexing in general. So I wanted to give you guys some more information about our flexible antibody labeling kits. So our flexible kits allow for easy antibody labeling using a high affinity flex linker. And so what makes these kits so great is that it's compatible with any primary antibody. So it doesn't necessarily need to be used with a protein tech primary antibody. It can be an antibody from any manufacturer. Another nice thing about these kits is that it works with any antibody concentration and formulation. So oftentimes when you're doing, traditional methods of antibody conjugation, like chemical methods, you need to ensure that your antibody is in PBS only. And so you need to do some sort of buffer exchange. And during that process, you end up losing quite a bit of your antibody. So it becomes very expensive. But with the flexible kits, you can go ahead and use your antibody in the a buffer that it already is in. Um, I know Anandita covered this quite a bit, but one of the benefits of the flexible kits is that you can label as little as zero point 0.5 micrograms. So you can label just as little as you need, or you can scale up if you need to. So basically, the idea is that you don't need to devote an entire tube of your antibody if you're only going to do an experiment a few times, or you want to use other conjugates with that same tube. It's also a very fast and easy protocol. So the protocol for labeling your antibody only takes 10 minutes. And the way that it works, as you can see in this figure here on the bottom, is you basically have your primary antibody. You then introduce your flex linker, and your flex linker has the conjugates covalently conjugated onto it. So within minutes, your flex linker is going to bind to the FC portion of your primary antibody. And then you just add in a flex quencher, and that's going to bind to any of the unbound flex linker, and then that's it. So then your antibody is labeled and ready to use. And because you are pre-conjugating your primary antibodies, you can multiplex using same species antibodies. So if all your favorite antibodies are in rabbit or mouse IgG1, you can use them as long as you have pre-labeled them with the flexible kit. The kits are also validated in multiple applications, so you can use it for IF, flow cytometry, or Western blot. And we do offer our kits as the original Flexible, but we also have a newly launched Flexible 2.0. So Flexible 2.0 has been optimized for immunofluorescence. So basically, it's the same exact thing as our original Flexible, except it has more dyes on the Flex linker. And so it's going to be ideal for kind of those low abundance targets and just for IF. So right now we offer these kits in these specificities shown here on the right and these conjugates. I also just want to show this comparison figure. So this comparison figure is showing the differences in staining when using antibodies that are labeled with Flexible 2.0, our original Flexible, or primary and secondary antibodies. So each column here is going to be showing you a different target. And on the top, you can see the primary antibody species and the specific IgG subclass. So overall, what this figure is showing us is that the original flexible acts pretty similarly to what you would expect from a pre-conjugated antibody, where the signal is going to be a little dimmer than if you were to use a traditional, primary, secondary, and go that route. However, Flexible 2.0 not only performs just as well as using a primary and secondary, but for certain targets, you can see that the signal was even brighter. However, if you are looking at, more popular and high abundance targets for IF or flow cytometry, Protein Tech also offers primary antibodies that are pre-conjugated to our Coralite dyes. So our in-house Coralite dyes are bright fluorescent dyes that deliver high photostability and minimal fluorescent bleed through, which makes it easy to combine 2 or more dyes in one experiment. And our Coralite dyes do have the equivalent brightness to Alexa Fluor dyes, and our antibodies are available in various conjugates, including 405, 488, 555, 594, 647, and 750. And lastly, I do want to highlight a couple of our secondary antibodies for traditional IF. So here I'm highlighting our nanosecondary reagents, which are nanobodies that are conjugated to Alexa Fluor or Coralite dyes. Our nanosecondaries are recombinantly manufactured and have a defined degree of labeling, which is going to be ideal for reproducibility across your experiments. They also have a high affinity for binding to primary IgG antibodies in a subclass specific manner, which will ultimately result in cleaner images. So you can see an example of this on the very right where you see that blot. And you can see that each nanosecondary that was tested only recognizes one subclass of IgG compared to the traditional rabbit secondary shown at the bottom. That was from a competitor, which recognizes rabbit IgG, but also some other subclasses of mouse IgGs. Also, since nanobodies are so small, they are about 10 times smaller than traditional antibodies, they do have the ability to penetrate tissue better for more even staining and can bind very closely to your protein of interest. And this is going to be great for generating high-res images, using, confocal or super resolution microscopy, which you can see in this image here in the middle, looking at vimentin in HeLa cells. And very lastly, I just want to highlight our multi-Rab goat recombinant secondary antibodies. These secondaries are a multiclonal mixture of recombinant monoclonal antibodies which recognize multiple epitopes on the same IgG. So the idea behind this product was to generate a secondary that had the high sensitivity of polyclonals with the superior specificity of monoclonals. Because we are incorporating the recombinant technology, our multi-Rabs have minimal cross-reactivity, unlimited supply, and high lot-to-lot consistency, making them ideal for reproducibility and your long-term projects. So as an attendee of today's webinar, you will receive 2 follow-up emails. The first one will have a recording of this webinar, as well as a link to request a free sample of one of our Flexible 2.0 kits from a selected list. And then the second email will be a certificate of attendance for this workshop. If you want to go ahead and claim your free sample now, you can also just scan the QR code and fill out the form now, but we will also send a link in the email too. And we do ask that you please allow 1 week for that follow-up email to be sent. So with that, I think we can go ahead and begin the Q&A. If after the Q&A at any point you guys have any questions for us, you can always email us at events@ptglab.com, or you can also reach out directly to Aanandita and Ola. And if you liked today's event and want to learn more about any upcoming events we do follow our social media because we post about all our upcoming webinars there. So with that, I will go ahead and begin the Q&A. We have quite a few questions. So the first question was asking about, when you're using 11 antibodies, do you see any aggregation of antibodies in the sample?
So maybe I will just start and Aanandita can follow up on that. So no, I have not experienced, me personally, any aggregation of the antibodies. What can be experienced is that at some point, at like going further in rounds, you can start seeing some leftover signal, some background signal, but the aggregation per se is never the case, and the signal is usually nicely, any aggregations that could be perhaps there are removed in the washing process, bleaching process, etc.
Yeah, I agree. I think it also helps if you ensure that your previous sample has been bleached before you move on, and also potentially on how strong your antibodies are, but usually it will get rid of it. We also varied our antibodies quite a bit, so we weren't labeling the same cell that many times. So potentially if you did the same cell 5, 6 times, you might see some buildup, but you tried to vary it between the different retinal cell types. So maybe that was also a contributing factor, but if you ensure the bleaching, hopefully that shouldn't cause any aggregation.
That kind of leads pretty well to this next question. So how many rounds of bleaching and staining can be performed before the signal degradation affects the data quality?
In my experience, it was 4 to 5 rounds. I don't know if Ola's experience has been different. I know the original IBEX paper did a lot more rounds, but they were also using mouse tissue. So the size and thickness of tissue varies quite considerably. Zebrafish retina sections are obviously much tinier. So it might depend on your sample, it might depend on how thick you want to actually slice your sections, because ours are pretty thin. I think we do 10 micrometers. So it can depend on that. And also how long your tissue actually stays on the sample. But theoretically, that 4 to 5 is how much, with 3 antibodies per round, is how much we found usually.
I had the same experience. I think I got into 5 rounds, but we know that definitely more is possible. We have a neighboring lab that has done, I think, up to 60 antibodies altogether. So as Aanandita said, it depends on but that was human sample. So it all depends on the sample you're working with, and if you even need 60 antibodies, maybe if you work in immunology lab, you do. but we usually get into the 5 rounds, which can already give us a lot of information.
Great, thanks. I'm glad you guys mentioned about kind of the preparation of the tissue and like the thickness, because we did have a couple questions asking, ideally, is IBEX better for thicker sections, whole mount, or is it generally used in thinner sections?
I don't think we've tried too much varying the thickness of when we do sections. I know Ola usually, just because it is adult tissue with killifish, does sections that are a little bit higher than what we do for zebrafish. So there might be specific differences. With whole mount, it is obviously very different. I think the tissue can last a little better just because there is more of it, but then you have different challenges with whole mount because with like mounting issues and the penetration of the antibodies. So for more details on that, we've done whole troubleshooting in our paper. But yeah.
Great. We had a couple questions here asking what fixation protocols work best with IBEX for maintaining tissue morphology and antigenicity.
So except for the, after we pre-coat the slide with chromalum gelatin, we do not do any fixation afterwards. Of course, while we call the animals, when we collect the samples, they are fixed in 4% PFA and then sectioned, dehydrated in sucrose. But after that, we don't do any extra fixation steps. What I could recommend, what I think I tried at some point, is to, before the first round of immunostaining, give it extra 15-minute fix with 4% PFA, but we don't usually do that. We didn't find it to help much with the tissue staying on the slide, and the fixation in PFA can alter your signal a little bit, the way your antibodies look like. So we don't necessarily recommend that.
Great, thanks. There have been a couple questions asking specifically about the fluorophores. So whether there are certain fluorophores that you guys were unable to bleach, or if there are certain fluorophores that are resistant to the bleaching process.
Yes, I think as I mentioned during the talk, we did have trouble with Alexa 555 as well as Alexa 594. And that is particularly when we're using secondary antibodies. But I think there's also a couple of factors that play into it, which is the strength of the antibody you're using. If you're looking at an antibody against a protein that's very highly expressed in your tissue sample, you might have trouble anyway, but particularly in 555 and 594, we did notice issues, which is why we then turned to antigen retrieval. So if you're having trouble with any of these channels, I would recommend trying a gentle antigen retrieval. So we just put some sodium citrate on the slide, enough to make sure it doesn't get dehydrated, put it in the oven and leave it at 60 degrees Celsius for 20, 30 minutes. And that really helps with reducing the signal. We did not have issues with coralite antibodies, as far as I can remember. I think the only one we haven't tried is potentially 405 because we always had DAPI in that channel. But for all the others, I don't think we've had too much trouble. But then we usually tried to put our weakest antibodies in that channel anyway, because the original IBEX paper found that it was hard to bleach. So yeah, we may not have encountered the combination that would not bleach.
And kind of going back to when you were talking about using antigen retrieval, there was a question asking about whether antigen retrieval is required after doing the in situ hybridization, or can they just go ahead and do the IF right after?
So you don't require it. It was only because the way that we do HCR, we buy the amplifiers from the company that provides them the molecular instruments, and they use Alexa floors. So we can't use a different floor. But if you have a different amplifier that has a different fluorophore and your sample bleaches completely with lithium borohydride, go ahead. You don't need to do antigen retrieval.
Great, thank you. Let's move on to the next question. So there were a couple questions asking whether this process could be done in cells rather than in tissues sections.
Absolutely. I don't see why not. It has been done so far on regular sections, whole mount, flat mount. So I'm sure it's also possible on cells. Our lab has not done that. We are not working with cell culture, so we did not have an opportunity to test it. But I don't see why not. It shouldn't be working. I think it's worth a try, and finding out whether it'd be a good method for cell culture.
Yeah, I think it would just require me some optimization, like things like how long it takes to bleach. Maybe the cells are damaged quicker than the ones that we use, or maybe they require longer bleaching times depending on how thick your tissue is. And like all the different steps we've given particular times for, you might need to play around with that a little bit to adapt it. But yeah, I just want to see why not.
Great. Thank you. There is a question about the flexible kits, so I will go ahead and answer this one. There's a question about how long you can store, then use the flexible labeled antibodies. So we do recommend that once you have labeled the antibodies to use them right away. That's why, you can label just as much as you need. It's just a 10-minute protocol. So fresh is going to always be better. The kits themselves will last, but the conjugation, we do recommend doing it when you're ready to use. So the next question that we have here is about the bleaching solution. They were curious about how long is the bleaching solution active, and do you need to need a fresh batch for every round?
Yeah,
Yes,
good
you do need a fresh
question.
batch. Go ahead.
Good. Yeah, that's a good question. Yes, you do have to make it fresh every time you prepare it. So you have to dissolve it in water every time you want to do your bleaching, and wait up to 20 minutes for it to activate. How long does it stay active? I have no idea. I usually use it within 30 minutes. I saw there was also a question that 15 minutes is not enough for bleaching. What can I do? What we sometimes do is that after 10 minutes or 20 minutes, we add a fresh lithium borohydrate. We refresh it. Maybe this will help. But basically, I don't know how long it's staying stable for, because I never risk it. I just usually make a fresh one every time I want to use it, and try to use it, I would say, within 1 hour maximum.
Yeah, so I guess when the bleach is activated, you can see bubbles, as Ola mentioned. So you probably won't use up your whole sample. So if the one in your tube is still bubbled, I'm assuming the one on your slide is still active. When the bubbles disappear, I would use it right away as soon as you notice that it has been activated, and not stretch how much, but definitely between different days, I would use fresh solution.
Great, thank you. And kind of on the topic of the bleaching solution, there is a question asking what concentration you guys use.
So we use, I think, 1 mg per ml.
Okay, great.
But more details in the paper. So you can check the methods, and that should all be in there with the product, like where we got it from and everything.
Perfect. And then should all the rounds be done within 1 day, or can the sections be sorted overnight following cover slip removal?
Yes, you can. Obviously, we don't do all of this in 1 day. It's usually done over a whole week. So no rush with all of this. The way I would usually do it is if I did the first round of secondary antibody, I would then do it the day before, wash off secondary antibody, image it, and then do the bleaching in the afternoon. Once it's bleached, I would then, once you ensure that it has successfully bleached, apply the next round of staining, and then leave that with the flexible kit staining overnight, and then image it the next morning. But yeah, so it takes sequentially across a few days. You could do it one after the other if you wanted, but we just found that the staining works best when you leave it overnight.
Awesome. Well, thank you guys. Do you guys have any last comments? I think we're at the top of the hour. But we have some great questions that we did not get to. So I will just really quickly put up Aanandita's and Ola's emails once again, just in case you guys want to take note of that and send them off an email. I love that we had so many great questions today, such an engaging audience. So that's awesome. But here's that information for you guys. So I just wanted to thank everyone for joining today's webinar. And then, of course, Ola and Aanandita, this has been such a great talk, so much great information that you were able to provide for our audience. So thank you guys for being here. So with that, I will go ahead and end the webinar, and I hope everyone has a great day or night, depending on where you are tuning in from the world. Take care.